my name is Erin Conley, and over the past 3 years, I have had the privilege of working under the direction of Dr. Kate Cooper and studying noise-induced hearing loss in mice possessing the plus over v-2J Cadherin-23 genotype. Now, I know that this is a lot to take in at first, so we'll go ahead here and break it down together. Firstly, noise-induced hearing loss, just as it sounds, is hearing loss that has been caused by repeated or long-term exposure to loud noise. Now, regarding this plus over v-2J Cadherin-23 genotype, it's first important to understand what a genotype is. And a genotype is comprised of two alleles, which are essentially two halves of a set of instructions of which you get one copy from each of your parents in order to create a complete set. Now, genotypes code for many things, such as hair color, eye color, height, blood type, you name it. These genotypes are decided from your genes, which are specific sequences that tell your body to do or make certain things. Now, for example, imagine we have the gene that tells my body how much pigment to make in my eye, resulting in which eye color I have. Now, say this first one here tells my body that I have blue eyes, and the second one causes for my body to create the pigment for brown eyes. Now, this plus over v-2J genotype is specifically relating to this gene called CDH23 that is important for Cadherin-23 protein production. The plus is indicating a normal allele, or what we would expect in a healthy individual, and in this instance, signifying normal Cadherin-23 protein levels. Whereas the v-2J allele causes for what research has shown to be a null mutation, meaning that Cadherin-23 protein is not made well, or that something is wrong with its structure, resulting in it not having a very good stability. So now you're probably wondering, so what? And how is this Cadherin-23 protein connected to hearing? Well, it's related to your inner ear and the hearing structures that reside within it. As you can see here, you have your inner ear, and this little spiral structure is called your cochlea. Well, within your cochlea, you have these hair cells, and on top of these hair cells are these structures called stereocilia. And these stereocilia are organized in a stair-like structure, and they are connected via these tiny structures as shown as blue lines here, called tip links, which are comprised of this Cadherin-23 protein. Now, these tip links are responsible for detecting when these stereocilia bend back and forth due to sound waves, resulting in a signal that is sent to the brain, ultimately giving us our ability to hear. Now, if this Cadherin-23 protein has a faulty structure, as with the v-2J allele, the tip links tend to be more brittle and break easier. And without having any tip links, there's no way for the stereocilia to detect movement, and therefore there results in no signal, and we lose our ability to hear. This ultimately led us to our research questions of, will the added stress of noise damage increase the susceptibility and hearing loss in these mice with the plus over v-2J genotype, as well as does one plus or normal or functioning allele offer enough protection to lessen the effects of noise-induced hearing loss, with our big picture goal of being to better understand noise-induced hearing loss in order to hopefully better combat and or prevent it. As I had mentioned before, currently damage is irreversible, and therefore hopefully gaining knowledge to see if we aren't able to medically repair these damaged structures in the future and having the ability to gift people their hearing back. So in order to test this, we had conducted many hearing tests that we did before and after noise damage in order to see if the hearing had changed as a result of the noise itself. And what we had found was no significant difference in hearing between the genotypes that we had tested, leading us to conclude that the plus over v-2J was not more nor less susceptible to noise-induced hearing loss when compared to the other genotypes that we had tested. And while our results aren't exactly what we were anticipating, there are some limitations that we identified that may offer some insight as to why we had received the results we did. The first one being that we had a very small sample size, as working with live animals is hard, and it's always very unpredictable as to what we will be fortunate enough to work with. And secondly, the age of the mice. And while our specific research is relating to noise-induced hearing loss, trying to find the perfect age at which the mice aren't so young that they are resistant to the noise-induced hearing loss, but also not too old that age-induced hearing loss is going to take precedence in our results, is a tricky middle ground to try and find. All of these things are things that we can continue to manipulate in future research to hopefully get better results and gain a better understanding of noise-induced hearing loss to help those who are affected.